Swayam Prabha. Digital India. Educated India.
Welcome back to the lecture series in animal physiology. We are into the 11th week and we are starting the third lecture of 11th week. So today as we promised each other, that we'll be talking a little bit more about the histology of the blood vessels which are traveling, coming from the heart and coming back to the heart, and in that process there are several changes which are taking place. So let's start directly into it, and then we'll talk about the capillaries and the exchange of the real exchange bed, where there are a lot of pressure differences, a lot of cross-sectional differences, and we'll enumerate all of that. Okay. So let's start off with our architecture of the blood vessels. Okay, this is week 11, lecture 3. Now, when we talk about the vessel which is coming out from the heart, which is the aorta, this is called elastic arteries. These are extremely elastic in nature, as I was telling you. Elastic, and if you look at that elastic structure, they will be fairly thick and something like this. Tunica interna, tunica media, and tunica externa, and they can withstand huge amount of pressure. Okay, so this is tunica externa, tunica media, tunica interna, and in between, of course, you have external elastic layer and internal elastic layer, what I have already shown you. Okay, see this one, which is the endothelial lining, and here you have the internal elastic layer, you have the external elastic layer. This is where you have the endothelial lining, and here you are having internal elastic layer, and here you have the external elastic layer, and these are called elastic arteries, which includes aorta. So now the diameter of it kind of narrows down. The next level, what you see is this. Which is called your muscular artery. You have more smooth muscle in them, and of course again same color coding if you follow. This is your endothelial lining. This is where the real blood cells are kind of hitting upon. Then you have internal elastic layer, and you have this external elastic layer. So this is the second level of artery. So in terms of the diameter of the vessel, if you look at it, as I was trying to tell you in the last class, so the diameter of the vessel is now reducing. So, okay. Now, this becomes even further narrowing down. Now, that is what you are going to see now is called arterioles. That's it. So, you have essentially what you have here is smooth muscle cells, that is tunica media, smooth muscle cell, tunica media, and you have the crippled endothelial lining, and you have the basement membrane on which the endothelial lining is sitting. So, the thickness of the vessel is slowly decreasing further. Okay. Now, arterioles become even further narrowing down to something called continuous capillaries. This continuous capillaries is even much narrower. Yet these continuous capillaries, they are purely made up of the endothelial lining, okay, and these continuous capillaries have lot of structures called fenestrae. Fenestrae are the windows where exchange of gases and other particles takes place. So, this is the only place where the integrity of the structure is kind of being compromised big time. So, this is the narrowing which is happening. This is the picture I wanted to show you. And out here, there is another thing which happened. There are a lot of branching which will happen here. Enormous amount of branching, and there is a significance to this branching. We will come to that soon. It's kind of branching which will take place here. And if you remember, recollect the picture I showed you or I drew in the last class, where I told you in the lungs this is the kind of exchange, and in the other tissues the exchange is different. So, it is something like this. Okay. This is where the vessel is now narrowing down big time. Now, from here the vessel, so this is where the oxygen is being given. Now, all the tissues which are surrounding, they are picked. So, let us denote the tissues with green. The one which are metabolizing, so they are taking the oxygen. Now, these same tissues give away the carbon dioxide, and we will start to indicate it like this. The structure is now becoming again merging at one point. Okay. So this merging, so the structure out here is called fenestrated capillaries. So most of these are called, so these capillaries, if you look at it, if this is the capillaries, so the fenestration increases even further out here. It's called capillaries followed by fenestrated capillaries. These fenestrated capillaries now pull together, and you have seen arteriole. Now you'll be seeing the diameter is now increasing of these vessels as compared to their arteriole is called venule. Now the venules become further enlarged to be called as, so this is not by scale, but just you have to understand, like medium sized veins followed by large veins with decently good amount of cross sectional area through which the blood can flow. And this is where your superior and inferior vena cava comes into play. So this is how our closed loop structure really looks like. And very interestingly, these structures have one-way valve. Because the pressure with which the blood is being pumped here, this is, we'll come to this, maximum pressure. And by the time the exchange and everything is taking place out in the fenestrated capillaries, the pressure is dropping. Yet the blood has to come back to the heart. See, in order to ensure that there is no reverse flow of blood, veins are equipped with structures or valves, you can call them, preventing backflow, whereas arteries does not need that. Okay. So this is the overall architectural difference or architectural changes what you will be observing. And I'll take a little bit of a detour to just give a little bit more detail about this fenestrated structure. So these fenestrated structures are something like this. These vessels are, if you see the architecture very clearly, at this level where the fenestrations are, the vessel architecture is more like this. We have talked about the endothelial cells. So this is, imagine this is the endothelial lining. This is the whole vessel. So let me just make the, if this is how the vessel looks like. Okay, so you'll see the gaps between the endothelial lining, and this is what I was telling you about fenestrated capillaries. And going by the color code, these are the endothelial lining. okay, and the blood flows through this tube, and this is where the exchange of oxygen and collection of carbon dioxide taking place. And this is what are those wonderful, what we call them as windows of the capillaries, making them fenestrated capillaries. Okay, so this basic understanding is exceptionally essential. But then, having said this, I will introduce another word which is called arteriosclerosis. You must have heard this word at some point or other. What is arteriosclerosis? Arteriosclerosis is the thickening and toughening of the arterial walls. Okay. The condition may not sound life threatening. It is not really a life threatening in the beginning, but complication related to arteriosclerosis account for roughly huge amount of death. Okay. So, the way it works is, say for example, this is the cross section So, out here, if this is the cross section, this is your tunica, externa, media, interna and all that stuff, and here you have the endothelial lining. So, now on top of endothelial lining you are having depositions. So, once you have depositions around it, your amount of blood flow plus pressure on the vessels increases, and that may lead to, at times may lead to damage, or sometime even may lead to something called aneurysm, where part of the capillaries or part of the vessels does something bizarre. It is just a part of the vessel all of a sudden become twisted like this. This is a classic case of aneurysm, and this aneurysm may lead to the damage of the vessel because of unusual kind of movement the blood has to go through. Those who does fluid dynamics, they will be able to appreciate it better, that if something is flowing like this and all of a sudden it has to take a very peculiar path like this because of some XYZ pressure or growth on the vessel kind of takes. So, these are some of the things which are commonly being observed in the arteries. And having said this, now you might wonder that why it is challenging to take blood from the arteries. You really need a huge amount of skill set before you can target to take blood from the arteries, because they have a huge amount of pressure with which blood is flowing. Unless otherwise it is needed, doctors prefer to take the blood or puncture on the blood collections from the veins. instead of hitting onto the arteries. This is something which should strike you, that why they look for the veins. Okay. So, with this I will close in on this class. In the next class we will talk about how the pressure and how all these differences are being calibrated. Okay. Thank you.